Yes, thank you very much for having me here today. Ella, as introduced before, I'm a PhD student in the Neuropsychopharmacology research group led by Dr. Chris Longtote. So today I'm going to be highlighting one of our current project related to mitochondrial dysfunction in Alzheimer's disease patients. I have no conflicts of interest to declare. So at the Neuropsychopharmacology research group, our goal is to optimize. Thank you very much.
This is the difference between supervisor and it's much different.
Thank you. Thank you. So at the Neuropsychopharmacology group, our goal is to optimize interventions for neuropsychiatric and cognitive symptoms associated with medical illnesses. So in our clinical trials, related to medications, modulation, exercise, we're looking into clinical factors and biomarkers of interest for mitochondrial dysfunction, oxidative stress, inflammation, and neuroimaging. For today, I'll be highlighting a project looking at the preliminary work from blood biomarker of mitochondrial distress. So in Alzheimer's disease, which accounts for 60 to 70% of all dementia cases, describing the progressive cognitive decline that can impact daily function. MCI, which is mild cognitive impairment, describes a phase between normal aging and AD when the memory decline has just begun with minimal or no functional deficits. So early disruptions in episodic memory, particularly recall memory, are characteristic hallmark symptoms in the early stages of AD. And there's currently lack of accessible and effective treatments for early AD. Disease-modifying monoclonal antibody treatments are recently approved in the US, aducanumab and lecanemab, but they're expensive and require intravenous infusion, making them not accessible to many. Side effects are also of concern with the risk of amyloid-related imaging abnormalities and brain hemorrhage. There is a need to investigate new markers and targets for development of novel therapies. So here comes in the mitochondrial dysfunction, which can be a key contributor to the pathogenesis of AD. CCF mtDNA are fragments of mitochondrial DNA released from cells as a result of mitochondrial dysfunction. Brain CCF mtDNA has shown potential as a biomarker for neurodegenerative diseases in the early stages. Somatic mitochondrial DNA mutation progressively increase with age, but are higher in AD brains and may contribute to AD development. We also see reduced CSF circulating cell-free mitochondrial DNA in neurological conditions. Limited research is available on blood or peripheral CCF mtDNA in MCI, particularly, and also mild AD patients, which would be a more accessible measure. Thus, I examined the serum CCF mtDNA levels and recall memory performance in MCI and mild AD patients to enhance our understanding of this relationship with cognitive decline. So, quick overview of the methods. We look at our patients in clinically diagnosed MCI and mild AD in the EXACT study with a 5-week study for exercise and TDCS. Just looking at the baseline level for now, I look at the association between the log CCF mtDNA levels and recall performance using multiple linear regression, controlling for covariates age and physical activity. The CCF mtDNA is measured by Dr. Anna Andreas' lab using qPCR with primer for the MTND4 gene. So here are some of the preliminary results with the sample of 29 participants. We have considerably even split between sexes with 59% male, predominantly white, high education level, and the majority, 72% are MCI. Our CCF mtDNA levels were associated with worse recall performance with LTQ, which is physical activity, and age. There's a partial correlation plot right there. Controlling for LTQ and age, 1 log CCF mtDNA unit was associated with 1.87 fewer words recalled at a median effect size. So the takeaway from this preliminary work is that higher serum CCF and mtDNA concentrations were associated with worse recall. The finding supports an association between mitochondrial dysfunction and cognitive function in early Alzheimer's disease. There are several limitations. First, the sample size is small. We will be going for 60 participants in the exact study, so we will anticipate having more sample to look at. Also, there seems to be a high variability in the CCF, MTDA, and serum level between individuals. And also, future research could compare healthy controls with MCI mild AD patients to evaluate the diagnostic capacity of the biomarker, and also longitudinal changes in response to different interventions. In this study, which I hope we'll have a chance to explore upon completion, looking at its response to exercise and brain stimulation. Also, we could also examine other mitochondrial dysfunction markers to better characterize the underlying mechanism. Thank you. Here are my acknowledgements.